Okay, so we're going to talk about how the cell is able to direct proteins for degradation in the lysosome, and also how it's able to recycle damaged or non-functional organelles. So the lysosome is involved in degradation of many macromolecules. The process begins when you have some proteins, such as these blue proteins here, on the membrane of the cell, and they have to be degraded. So they go through the process of endocytosis, where you have formation of the clathrin AP2 complex on this budding vesicle. Then you have the dynamine hydrolyzes the GTP to pinch the vesicle into the cytosol. Once the vesicle is in the cytosol, the protein coat along with the clathrin coat is disassembled, and now you have this vesicle with the proteins that are bound to be degraded. And this is known as the early endosome. At the same time, you have proteins that are meant to be transported to the lysosome to operate as lysosomal uh, specific enzymes that either degrade other macromolecules or are involved in the pumping of protons and other ions across the lysosome membrane, shown here in green. These are transported to a late endosome through the formation of this clathrin AP1 complex on the around the membrane from the trans Golgi network. Then you have the disassembly of the coat, and then these lysosomal pump membrane proteins are in green. They fuse with the late endosome, and now you have a mixture in the late endosome of blue and green proteins, which correspond with the blue, which are to be degraded, and the green, which will function in the lysosome. And so the lysosome or the endosome has this process of organizing these proteins that are to be degraded in blue from the ones that are to be functioning in lysosome. And so the way it does it is it forms this inner endosomal budding, where the proteins that are included in these uh, into these vesicles are only the ones that are to be degraded, so blue. And this forms basically a multi-vesicle endosome, which once all the degraded once all the proteins that are to be degraded are budded into the endosome, the endosome will bind to the it'll fuse with the lysosome, and the green proteins are will become part of the outer or part of the lysosome membrane, while all these inner vesicles will basically be within the lysosome, and they'll be broken down into their monomers. And so the way that the endosome will process these proteins that are to be degraded, it uses this um, HRS proteins, and also these cargo proteins that are to be degraded in blue. Notice they're all ubiquitinated, so they have ubiquitin attached to them. And what you have here is you'll have the bud that forms on this endosome toward the interior, towards the lumen of the endosome, and these HRS proteins will organize this ESCRT complex assembly, which will help the vesicle form. Right, it helps it pinch off into the lumen of the endosome, and then after that happens, a VPS4 protein will drive the disassembly of this complex through ATP hydrolysis. And it turns out that some viruses like HIV use a similar method of virus particle budding. They have this HIV gag protein, which has ubiquitin attached to it, and it basically resembles this HRS protein on the endosome with ubiquitin. And so it uses this gag protein to basically recruit these C or ESCRT assembly proteins on the forming HIV envelope. And so once this assembly has been made, it'll help the HIV envelope bud off of the plasma membrane, and then you get a free HIV particle. And then of course the disassembly of this complex would be driven by VPS4 through ATP hydrolysis. And from these images we could see that in a normal cell you have budding of the HIV capsule, and then the actual complete pinching off of the completed HIV virus through this complex here, the ESCRT assembly. And in cells with mutations in either the proteins that are part of this ESCRT assembly, or also the VPS4 protein, you have this situation where the HIV envelopes will form on the surface of the cell, but they will not completely bud off and become a particle. They form these chains of partially completed viruses that are non-functional. And also there's another idea here where the lysosome is able to degrade full organelles such as mitochondria. And so how this happens is during periods of autophagy or self-eating, you have formation of these cup-shaped membranes, which will form next to some organelle, such as this mitochondrion, that might be defective, and they basically grow. The membrane will grow until it fully encapsulates it in a double membrane structure, and you have these proteins on the outer part of the membrane called the ATG8, and they'll basically help the with the fusion into the lysosome, after which the lysosome will, the outer membrane of this double membrane autophagosome will become part of the lysosome, and the inner membrane will just be included inside of this vesicle in the lysosome. And of course the lysosome has proteins that will degrade any components inside of this vesicle into amino acids. And you also have situations where this cup-shaped membrane will nucleate around like some random part of the cytosol, and it'll basically return to the lysosome uh, some some empty uh, material basically.